In this lecture, we will examine gluconeogenesis. This is a biochemical pathway for synthesis of glucose. Gluconeogenesis is a biochemical pathway for synthesis of glucose from pyruvate. Now, glucose could be synthesized also from non-carbohydrate precursors. So main non-carbohydrate precursors are lactate, which is a result of lactic acid fermentation, which we just covered in the previous lecture. Also amino acids, which are derived from proteins, and during starvation, they could originate from breakdown of proteins in the skeletal muscles. So amino acids could be dietary amino acids from proteins that were consumed, or during starvation from breakdown of skeletal muscles. Another common precursor would be glycerol, which is a result of hydrolysis of triglycerides or fats. Glycerol can be either metabolized by glycolysis to produce energy, or it can enter gluconeogenesis to produce glucose. All these non-carbohydrate precursors are converted into pyruvate and then enter gluconeogenesis, or can enter gluconeogenesis at a later stage. In the course of glycolysis, glucose is converted into pyruvate and energy. And in the course of gluconeogenesis, pyruvate and energy are converted into glucose. So in that sense, the two pathways are opposing pathways. But the two pathways are not reversals of each other. That means that one is not reverse of the other pathway. The reason for it is that glycolysis involves three irreversible steps. And those three steps are catalyzed by enzymes hexokinase, phosphofructokinase, and pyruvate kinase. Since those steps cannot occur in reverse, in gluconeogenesis those steps have to be bypassed by different steps. And so gluconeogenesis as biochemical pathway involves different steps that bypass these pathways. And so these would be the ways to bypass pathways. Phosphoenolpyruvate is formed from pyruvate by way of oxaloacetate, a new intermediate. Fructose 6-phosphate is formed from fructose 1,6 bisphosphate by hydrolysis of the phosphate. And finally, glucose is formed by hydrolysis of glucose 6-phosphate. The first stage of gluconeogenesis involves steps that are designed to bypass glycolysis steps catalyzed by pyruvate kinase. And that involves conversion of pyruvate into phosphoenolpyruvate. So pyruvate in a reaction with carbon dioxide, ATP, and water, catalyzed by enzyme pyruvate carboxylase, is carboxylated. So carboxylation means that molecule of carbon dioxide is added to it, and product is oxaloacetate. This conversion of pyruvate in phosphoenolpyruvate takes place in mitochondria. So first step, conversion of pyruvate into oxaloacetate actually takes place in mitochondria. And then in the second step, oxaloacetate is decarboxylated and phosphorylated. Enzyme is phosphoenolpyruvate carboxylase, and product is phosphoenolpyruvate. Reaction is decarboxylation of beta-ketoesters, and that's a well-known process from organic chemistry. If you recall organic chemistry, beta-ketoesters spontaneously decarboxylate, and mechanism is outlined here, to lose carbon dioxide and produce corresponding carbonyl compounds, as shown here, ketones. So, and in this second example, we have decarboxylation of beta-ketoacids, and product is corresponding carboxylic acid. In each case, decarboxylation actually initially produces corresponding enol, and actually it's that enol that is trapped as phosphate. So enol reacts with actually GTP, and product is corresponding phosphate, phosphoenolpyruvate. So the overall reaction is pyruvate reacts with ATP, molecule of ATP, molecule of GTP, and molecule of water. Products are phosphoenolpyruvate, molecules of ADP, GDP, inorganic phosphate, and 2 protons. Now, pyruvate carboxylase is a mitochondrial enzyme, while other gluconeogenesis enzymes are present in the cytoplasm. So, oxaloacetate must be transported into cytoplasm to complete the pathway. And to that end, oxaloacetate is first reduced to malate. And then malate is transported into cytoplasm, and then in the cytoplasm, malate is reoxidized back to oxaloacetate, and then this step provides NADH for use in gluconeogenesis. Formation of oxaloacetate and decarboxylation couples unfavorable reaction, and that's formation of phosphoenolpyruvate with favorable reaction, and that's decarboxylation. Phosphoenolpyruvate is next converted in a series of steps into fructose 1,6-bisphosphate. And those steps are reverse of glycolysis. All of those steps are reversible, so they occur in what you would consider forward fashion in glycolysis and reverse in gluconeogenesis, the way we have learned them, in the order we have learned them. So in this case, starting with phosphoenolpyruvate, through action of enzyme enolase, the phosphoenolpyruvate is hydrated, and then through action of phosphoglycerate mutase, 3-phosphoglycerate is obtained. In the next step, 3-phosphoglycerate, through action of corresponding kinase, is converted into 1,3-bisphosphoglycerate, and that requires 1 molecule of ATP for phosphorylation. And then in the third step, 1,3-bisphosphoglycerate is reduced to GAP, or glyceraldehyde 3-phosphate. In the next step, GAP that has been produced in the previous step undergoes isomerization. Only half of those GAP molecules, glyceraldehyde 3-phosphate molecules, undergo isomerization. The other half, of course, are unchanged because half is needed to prepare fructose 1,6-bisphosphate. So the half that is isomerized through action of triose phosphate isomerase into dihydroxyacetone phosphate. And then 1 molecule of dihydroxyacetone phosphate reacts with 1 molecule of GAP that has not been isomerized, and enzyme aldolase couples them into molecule of fructose 1,6-bisphosphate. And that completes this sequence. So the overall process is that 2 molecules of phosphoenolpyruvate have been converted into a molecule of fructose 1,6-bisphosphate. In the next step, fructose 1,6-bisphosphate is converted into fructose 6-phosphate, and that step is accomplished by action of specific enzyme, and that's fructose 1,6-bisphosphatase. So in this case, a reversible step of glycolysis is bypassed simply by the action of specific enzyme that is specific for fructose 1,6-bisphosphate. So it will only hydrolyze 1 phosphate group on fructose 1,6-bisphosphate, and product is fructose 6-phosphate. Finally, in the final stage of glycolysis, fructose 6-phosphate is isomerized into glucose 6-phosphate. And that's reverse of glycolysis. This series of steps, isomerization, is reverse of glycolysis. And actually, gluconeogenesis stops here. So once glucose 6-phosphate is formed, that's the end of gluconeogenesis. Now, glucose 6-phosphate could undergo a number of different transformations. It could participate in a number of different biochemical pathways. So glucose 6-phosphate cannot be transported out of the cell, and glucose 6-phosphatase, which converts glucose 6-phosphate into glucose so that glucose can be transported out of the cell, is regulated. This enzyme, glucose 6-phosphate, is present only in tissues whose metabolic function is to maintain blood glucose homeostasis, and that would be liver and to a lesser extent kidneys. Overall, gluconeogenesis is an energetically unfavorable process. It actually requires energy, and delta G for it is plus 84 kilojoules per mole, which means that it requires quite a bit of energy. And as you have seen, 2 molecules of pyruvate require also 2 molecules of ATP, 2 molecules of NADH, 2 protons, and 2 molecules of water to be converted into molecule of glucose. Also, 2 molecules of inorganic phosphate are released, 2 molecules of ADP, and 2 molecules of NAD. For additional NTP molecules, and that means 2 molecules of ATP and 2 molecules of GTP are needed to make this process favorable. And so when the overall reaction is calculated, the overall energetic balance for the entire process is calculated, then the overall process involving consumption of additional 4 molecules of nucleotide triphosphates, then the whole process is favorable by 38 kilojoules per mole, or exergonic by 38 kilojoules per mole.